Hi, my name is Masanori Sakaguchi. I'm from University of Tsukuba, IIIS. So today I'm going to introduce the research in our lab. We are conducting two different kind of research in our lab. The first is the basic research using rodent. How is memory processed during sleep? And the second, by using this mechanism that we found, we'd like to try the application of those to cure patients. Today, I'm going to talk about mostly about the basic research we are conducting. So, as you can imagine, memory is processed during sleep, and the sleep consists of two different stages. One is the non-REM sleep, and the second is the REM sleep. And there's a cycle of those non-REM and REM sleep during the night period. The REM sleep is basically found as frequently dreaming periods in humans first. And later, a lot of research has shown the function of the REM sleep. And for us, the most interesting phenomena is that the PTSD patients often suffer from nightmares about their traumatic memory. So it seems like the REM sleep has many functions for memory, such as memory consolidations or coping to the traumatic memory, to which sometimes some emotional reaction adjustment is required. And apparently, in PTSD patient, that is becoming too much, and the traumatic memory reaction continues. So we are particularly interested in the mechanism how the REM sleep is correlated with the reactivation of the memory. So in the rodent research, classic papers show that the neuronal activity patterns during the wake period re-emerge during the subsequent REM sleep. And other line of research has shown that sound stimulation during REM sleep, the sound which is associated with the face pictures during the wake period, this stimulation during REM sleep enhances the facial recognition. So we wonder what is the function of the neuronal reactivation during REM sleep, and actually how it is actually happening in the brain. So those type of episodic memory in which like when happens, what happens, and those kind of like informations we can declare or speak about it, those kind of memory is so-called episodic memory. And the episodic memory first processed in the hippocampus in the brain. So the hippocampus consists of several regions like a dentate gyrus or CA3 or CA1. But the dentate gyrus act as the first gateway of the sensory information which is received from the cortical areas. And those information is separated into different patterns and recognized as the different memory if those two kind of unit of the memory is similar. And this pattern separated information is sent to the CS3 region where this piece of the information is put back as one unit of the memory, and that's so-called pattern completion. And this pattern completed memory is sent finally to the CA1 region, where the several memory integrations happens, and the temporal storage, and also the sending back to the cortex of the information would make the memory endure more in a long term, so-called like it becomes a remote memory in the cortex. So this is a kind of like a closed circuit to process the memory. And we particularly interested in the dentate gyrus here. So in this dentate gyrus, the particular character of the dentate gyrus is that it holds so-called adult neurogenesis process. And in this adult neurogenesis process, new neurons are being generated throughout life. As you may know that in the mammalian brain, once the neurons die, they do not regenerate. However, particularly in this dentate gyrus adult neurogenesis system, the continuous neurogenesis make the new neurons every day, which are integrating into the memory circuit. And what's unique about it is that the young adult bone neurons, which is around 4 to 6 weeks age after the proliferation of the neural stem cell, they show enhanced excitability and synaptic plasticity. And that period is actually limited to those 4 to 6 weeks old period. So we targeted basically that young adult bone neurons because it is shown that the young adult bone neurons hold contextual informations, and that's actually very important for fear memory. So I'm going to introduce our key paper published recently about this adult bone neurons function in REM sleep for memory consolidation. So in this research, we use 3 different research techniques. The first is the so-called in vivo calcium imaging technique using miniaturized fluorescent microscope. So by doing this, we can specifically analyze the adult bone neurons activity in living mouse, which is under free moving condition. And the second is the so-called optogenetics. The optogenetics is basically a technique to remote control the target activity of neurons. In our case, we use to remote control, which means activation or silencing of the adult bone neurons activity during only REM sleep. And the third technique we utilized is the fear conditioning. So fear conditioning is so-called is associative memory. So we put the mouse into a specific chamber, then mouse recognize this chamber and create a special information or contextual information. Then the mouse received electric foot shock. After mouse is put back into the home cage, then during the 6 hours period, they associate the contextual information with the foot shock. So when the mouse put back into the context, then mouse recognize this context and show fearful response. In mouse case, it is actually so-called freezing, where mouse stop moving around. So we can quantify this freezing time period as an indicator for the memory strength. So first, we image the adult bone neurons activity in this mouse, which is going through the fear conditioning, training and test and consolidation. And we found that the adult bone neurons activity during the learning is reactivated during the subsequent REM sleep period. Second, to examine the causality of the activity during REM sleep for memory consolidation, we either activated or silenced the adult bone neurons activity specifically during the REM sleep. We found that both the activation and silencing impaired the memory consolidation, which is phenotypically observable as the reduction of the freezing level during the test. We also found that the silencing of the activity of the adult bone neurons during REM sleep elongated their spine neck. This means that the physiological activity of the adult bone neurons during REM sleep is essential for their structural plasticity of the adult bone neuron spines. So this is the experimental result using the calcium imaging and fear conditioning. So we divided this fear conditioning period into home cage, pre-shock period, and post-shock period, then the memory consolidation period in the home cage for around 5 hours, and we recorded the activity, particularly during the REM sleep and during the memory test. So mouse basically froze when we put mouse back to this test chamber. So we divided the adult bone neurons into two populations. The one is the REM active neurons, and another is REM inactive population. So this graph shows the percent active cell population. So let's say REM active neurons are 100%, then the only 25% or around that number of neurons activate during the pre-shock period. So REM inactive neurons, 25% also active during the home cage, and that goes on. And as you can see here, we found that the post-shock period, the REM active neurons, 50% of the population of the REM active adult bone neurons were active during the post-shock period, but only like 25% of the REM inactive neurons were active in the post-shock period. And there was a statistical difference here. On the right side, we are showing the same result in a different way. So we basically split the post-shock active neurons as 100% here, and around 60% of them is reactivating during the REM sleep, as you can see here. And also the post-shock inactive neurons, within that, around 25 to 30% of them are reactivating during the REM sleep. And there's a statistical difference here, which means that the post-shock active neurons, adult bone neurons, are reactivating during the REM sleep. So to show the causality of the activity during REM sleep, we did optogenetic silencing of adult bone neurons, specifically during REM sleep, within the memory consolidation period after the field conditioning. So first, we expressed the harodopsin, the optogenetic silencer, in the adult bone neurons using the same nesting Cree driver mouse, and we shine the orange laser during REM sleep. We use the control mouse in which do not express the harodopsin, but we shine the laser in the same way. And as you can see here, this is showing the percent time freezing during the test period. Compared with the control mouse, the mouse group, which adult bone neurons are silenced during REM sleep, shows less freezing level, indicating that the activity of the adult bone neurons during REM sleep is essential for memory consolidation. When we did the same thing after the memory consolidation period, we shined that orange light to the adult bone neurons during the 6 to 12 hours after the fear learning period. We did not find the difference we found in the figure here. So this means that the activity of the adult bone neurons after the memory consolidation is not essential for the memory consolidation. So we also found that when we silenced adult bone neurons activity during REM sleep, we found their spine neck length is elongated, as you can see in the red circles here. So we found many of these elongated spine in adult bone neurons dendrite. And so that is specific when we silenced adult bone neurons activity. So it means that the adult bone neurons activity during REM sleep is necessary for the physiological structural plasticity of the adult bone neurons spine. So then we did some gain of function study by randomly activate the adult bone neurons activity during the REM sleep. So we stimulated the channel rhodopsin 2 expressing adult bone neurons by shining blue light. We found that this 20 Hz random activation impairs the fear memory. which is phenotypically observable as the reduction of the freezing level. So this is a brief summary of the research. First, young adult neurons reactivated during REM sleep. And when we silenced adult neurons activity during REM sleep, as you saw, as a reduction of the freezing, impaired of the memory consolidation and the synaptic structures. And 20 Hz random activation during REM sleep also impaired memory consolidation. So these are our members, and so we are kind of like I'm very happy to work with these very talented young researchers. They are very diligent and friendly, and very versatile from many countries. And so we are like very lucky to work with many wonderful collaborators, and we appreciate the funding sources. Thank you so much for the listening.